Good morning, everyone. Today we will have a bioinformatics workshop series with title Basic Bioinformatics and Bacterial Long-Read Sequence Analysis Tools. Today, let me be your MC, and I will read the agenda. So we will, for the first agenda, we will sing our national anthem, and the second one, there will be like a welcoming speech from Mr. Budi Prawara if he was attend, and the next will be the material from Mr. Balamurugan Paryaswamy, and the last is the question and answer. Okay. Let's start with to sing our national anthem. Bariska, can you please play the music?
Mohon izin, saya tidak bisa share screen-nya.
Kayaknya harus di-unshare dulu, Mbak.
Mr. Balanya harus stop share screen dulu.
Yeah. Mr. Bala, can you please stop share screen? Yes, thank you. So, Bariska, please. Okay, let's sing our national anthem first.
Bentar, Bu.
Okay, just a minute. Maybe just wait a minute, it's technical problem. Okay, let's start.
Ada suaranya.
Nggak ada suaranya, Mbak. Mbak Riz. Maybe you can share voice, Mbak Rizka. Suaranya belum ada.
Ya, Mbak. Masih tidak terdengar ya?
Iya, nggak ada suaranya sama sekali.
Oh, mohon maaf semuanya. Sepertinya karena komputer saya ini, boleh di-share screen saja di Bu Maulida?
Boleh, ada linknya, Mbak.
Bentar.
BRIN, Inovasi Cooking Deodoran. Rasakan tetap dingin instan yang menjaga kunyah tetap kering dan wangi segar seharian. BRIN, Inovasi Cooking Deodoran. Rasakan bedanya.
Okay, the second term is Pak Rifki. We will have directly material
Pak
from
Budi juga.
Bala, or Pak Budi is
Yes,
already.
Budi is here.
Okay. So since Pak Budi is here, let us hear the welcome speech from Pak Budi, the head of the research organization for electronics and informatics. To Pak Budi, Prawara, time is yours.
Baik, terima kasih Bu Medina. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera. Very good morning. Yang saya hormati Kepala Pusat Riset Komputasi, Pak Rifki Sadikin, our distinguished speaker, Mr. Balamurugan Perias Wami from Oxford Nanopore Technologies, serta Bapak Ibu peserta workshop Bioinformatic di pagi hari ini. Pertama-tama marilah kita ucapkan puji syukur kehadirat Allah Subhanahu wa Ta'ala, Tuhan Yang Maha Esa, karena pada pagi hari ini kita semua diberikan kesehatan baik ya, sehingga kita bisa bersama-sama mengikuti acara workshop yang nanti akan disampaikan oleh speaker kita dari Oxford Nanopore Technologies. Ya, sebagai informasi ya kepada Bapak Ibu, bahwa salah satu fokus program Organisasi Riset Elektronika dan Informatika atau OREI itu adalah riset di bidang artificial intelligence dan big data untuk biodiversity. Nah, saya rasa kegiatan workshop di pagi hari ini adalah menjadi salah satu bagian dari fokus kegiatan riset kita ya, yang tadi sudah saya sampaikan untuk AI dan Big Data untuk biodiversity. Ya saat ini kita sudah memiliki beberapa fasilitas ya yang mungkin cukup lengkap, baik itu di ORE maupun di OR lainnya yang nantinya akan kita gunakan untuk kegiatan-kegiatan riset di bidang itu tadi ya, apa namanya, biodiversity, kaitannya dengan AI dan Big Data dan bioinformatika. Ya, sebagai contoh fasilitas HPC kemarin sudah kita tata kembali juga. Jadi sekarang ada 2 klaster, yaitu di Cibinong untuk klaster komputasi dan di Serpong untuk klaster GPU ya. Jadi 2 klaster ini kita harapkan bisa kita manfaatkan ya semaksimal mungkin. Saya rasa rekan-rekan periset dari bidang Bioinformatics yang tentunya nanti akan memanfaatkan juga ya. Nah, kegiatan-kegiatan semacam ini Pak Rifki yang mungkin nantinya akan kita perbanyak ya untuk meningkatkan kapasiti SDM periset kita ya, karena tanpa human capital ya, periset yang memang bisa menjalankan dan menguasai peralatan yang ada di kita, saya rasa riset-riset yang ke depan tentunya sudah menjadi tren yang diharapkan kita juga bisa berkontribusi itu tidak bisa berjalan dengan baik ya. Oleh karena itu saya mengucapkan terima kasih kepada Pak Rifki dan kawan-kawan yang sudah menyelenggarakan acara workshop ini. Dan untuk program kaitannya dengan riset di bidang genomik, misalkan ini ya workshop yang pertama ya Pak Rifki ya, yang di bakteri long read. Harapan kita nanti bisa kita gunakan untuk berbagai keperluan, bisa kita gunakan untuk pengungkapan sumber daya hayati kita, dan juga untuk berbagai keperluan yang lain seperti penemuan obat baru, medical, dan sebagainya. Kami juga di ORE, saya dan Pak Rifki, kita berusaha untuk bisa menjalin networking dengan pihak-pihak terkait ya, baik di dalam maupun di luar negeri, sehingga kita bisa memperkuat posisi kita di Indonesia maupun juga di kancah global ya, di bidang khususnya riset di bidang bioinformatik ini. Dan rencananya saya juga sudah berbicara dengan beberapa kepala OR terkait seperti OR kesehatan, bagaimana kita bisa bisa mulai jadi hasil dari workshop ini kan kalau tidak kita gunakan, tidak kita praktekkan, tentunya nantinya akan mubazir ya, sehingga kita coba mencari wadah kegiatan yang nantinya dapat rekan-rekan dapat mulai menggunakan apa yang sudah Bapak Ibu dapatkan ya selama ini maupun pada hari ini. Jadi sebagai contoh kami akan coba menginisiasi dengan OR kesehatan untuk bisa mulai mendevelop vaksin ya, vaksin untuk hewan. Ini sebagai tahap awal sebagai sarana kita untuk bisa mulai kita menggunakan fasilitas yang ada di BRIN, dan juga tentunya apa yang sudah kita dapat selama ini ya dari upaya untuk peningkatan kapasiti SDM kita juga. Ya, saya berharap kegiatan workshop pada hari ini bisa berjalan dengan baik dan bisa memberikan manfaat bagi kita semua. Demikian, sekaligus mungkin saya membuka acara workshop di pagi hari ini. Sekali lagi saya mengucapkan terima kasih kepada Bapak Ibu dan juga kepada rekan-rekan yang sudah membantu untuk menyelenggarakan acara workshop ini. Juga kepada Mbak Maulida ya sebagai moderator pada acara workshop ini. Terima kasih. Wassalamualaikum warahmatullahi wabarakatuh. Thank you very much.
Thank you, Pak Budi. Actually, I am not the moderator. Moderator will be delivered by Pak Rifki. I am just the MC. So, okay. Now let's continue with the next sessions. The lecture from Mr. Balamurugan Pariaswamy with the title Basic Bioinformatics and Bacteria Long Read Sequence Analysis Tools. For Mr. Bala, time is yours.
Okay, good morning, everyone. So, do you all hear me? Ricky, do you want to talk?
I think I should be introduce you first, then you can talk.
Go ahead.
Because I already wrote that the moderator is me, so so I think I will introduce you first to the audience, then you can start to talk. So thank you, Pak Budi, for the welcoming speech. So today we actually have an event for the workshop of bioinformatics. So this is the first time. Maybe we can have many series because we have many programs on sequencing, especially with the on technology, Oxford Nanopore technology. I will contact with Mary and you also what kind of another series for after this. But to have the background on the workshop, actually we have like a machine for sequencing at the genomic laboratory. We have long read technology like ONT, and also we have NGS and Sanger, and we would like to optimize also the computing environment at BRIN. We have this kind of specification right now, as mentioned by Pak Budi. We have a computational science clusters located in Cibinong, and this year we add like 19 plus not more for the computing nodes, and also we preparing the storage up to 6 petabyte, and I think we can optimize this system. And the plan is like we have kind of easy to use for the users to do repository or storing the sequence data, and also to do some analysis. But this is still ongoing. So let me introduce you, Mr. Balamurugan Pariaswamy. So I took from the link and also from the Google Scholar. Here he is right now is technical application scientist at Oxford Nanopore, but before that research scientist at the STAR Singapore research research center right there, and also a postdoc fellow at Genome Institute of Singapore. And Bala finished his PhD at 2012 at the ETH Zurich. Today the topics on the basics bioinformatics, and also maybe Bala will have some example from bacteria case. So we have, I think, up to you, Bala, or maybe 1 hour or 2 hours for the materials. Then after that, we have kind of question and answer. Maybe if there are some hands-on, if you want, then you can lead us to do some hands-on. So now time is yours. Please, Mr. Bala, to deliver your lectures or workshop. Thank you.
Thank you, Rifki, and thanks everyone for arranging such a wonderful webinar. It's a really cool idea. And I'm sure that we can do a lot many more of these kind of series, and every time we can introduce a module. And for example, based on your projects, be it bacteria this time, and maybe next time we can do human genome analysis and things like this. And we can. So today I have planned a little bit of an introduction because it covers basic bioinformatics, and then I will also show you practically some workflows. How do you really launch a workflow and analyze a workflow in the context of bacterial genomics, so that you will get a mix of some theory and some practicals. Obviously, we cannot cover a lot with the time frame that we have, but I'll try to give you a flavor of some of the basics in the theory, and also some examples, real-time examples, practical hands-on, and I will directly SSH into a machine and show you how to analyze some bacterial genome analysis and things like that. And later we can follow with some questions. So let me share my screen. Do you see my full screen? I hope you can see my full screen.
It's clear.
Okay, perfect. So let's start. Okay, so let me start with the section I have.
Yes, but it's not full screen.
Is it not full screen?
No,
Should be full screen. Yeah. So so I have broken this theory session into 4 parts. So basically, part 1, I would for beginners. I don't know, I assume that there are a mix of listeners here. So I would start with how we generate data, and what is Mino, for example, the basic sequencing software that you need to install on your machines if you want to do sequencing using Oxford Nanopore, and what is the larger technology behind it, how we generate data, and then the logics behind it. So a few slides on that. And after that, I will explain about the important files that get generated during sequencing, because for bioinformatics, the most important thing is one needs to have a good understanding of what are all the kind of files that get generated from a sequencing experiment. And then you have an idea of how to analyze the data. So I will introduce the data and file formats. And after that, I will give you pointers on some of the basics in command line. As it's a basic bioinformatics session, I'll just introduce some of the basics in command line. And later, I will follow up with all the different Oxford Nanopore solutions for bioinformatic data analysis, with a focus on bacterial genome analysis. And with that, I will end my presentation. And after that, what we can do is I can share my screen, and I will enter into a Promethean machine. And then I will show you in real time how to launch a bacterial genome assembly program. And maybe I will also show you some of the cloud-based analysis that we have given time permits. So with that, let's start. Okay, so Mino is our sequencing control software. And it does not only just sequencing control, but it does so many different things. So that's, as I said, it controls the entire sequencing. And when I say sequencing, that means actually generation of the raw electrical signal. As you know, in nanopore technology, when a DNA or an RNA passes through a nanopore, so there is a disruption in the electrical current, because there is a potential to the top and the bottom of the membrane. So when a DNA or an RNA passes through a small nanopore, the electrolytes get disrupted. So, and then that gives a squiggle pattern, which we call as, the disruption in electrical current we call as a squiggle pattern. So that is what we call as a raw signal. And that is what we also call as sequencing. And nanopore technology, unlike other third generation technologies, it is electronics based. So there is no scanner involved. So, and because of that, the machines are too smaller, because the chips are smaller. So the machine is smaller and it is portable. At the same time, it's accurate, and then also really, really fast. So the sequencing is actually raw data generation, and which is the recording of the raw electrical signal. And then the software Mino can convert this raw electrical signal by a process called base calling into nucleotide sequence. So once you have the nucleotide sequence is the starting point for many of the biologists. So basically, that is something where you can make sense out of it, and you can take the nucleotide sequence into downstream analysis. And then what else amino software can do? It can give you real-time quality control plots with which you can make some informative decisions on whether the run is going on well and things like that. And then it can also do some analysis in real time, for example, aligning against a particular reference and actually enriching for a certain target of your interest at the software level, or depleting a certain target of your interest at the software level. And we also have integrated end-to-end SARS-CoV-2 workflows plugged into Minnow. And in the future, we want to bring many different workflows directly into Minnow. So there will be one-stop solution so that you can not only sequence and control the sequencing device, but you can also do QC and you can also do analysis with one integrated software. So that is our future idea. But right now we have only SARS-CoV-2 plugged in into Minnow. But for other analysis, you have to use our other software solutions, which I will introduce in the slides to come. And finally, you will also get some QC reports in the form of HTML reports, which you can share with your collaborators. So that's the brief picture about Minnow. So Minnow is this essential software that is actually comes pre-installed on our devices, our sequencing devices like Mac1C and Promethean or a Gridiron. But if you're using a Mac 1B on your personal laptop or workstation, you have to download the Minnow software for the Mac 1B device onto your laptop and install it. And it is platform independent. I mean, you have a Minnow compatible software for Mac OS and Linux and also for Windows. So let's briefly go into base calling. So as I said, this signal that you see is this squiggle pattern that we are calling. This is the raw electrical signal disruption in the electrical current when a DNA or RNA passes through the nanopore, and that results in this conversion of this raw electrical signal into nucleotide information is what we call as base calling, and this is done by a software called Guppy. So this Guppy software is integrated into the sequencing control software, which we call as Minnow. So Minnow is actually a meta package. So in that meta package, there are several software programs all bundled together as a meta package. And then one software program, which is integrated into the larger sequencing software, which is the Minnow, is Guppy. And Guppy is the software that actually does the conversion of the raw electrical signal into the nucleotide information. And then there are several accuracy models. Guppy uses a neural network algorithm to actually translate this raw electrical signal into nucleotide information. And there are three different accuracy modes. So you have a higher accuracy mode, and you have a faster accuracy mode, and you have a super accurate mode. So for example, if you choose a super accurate mode, the raw read accuracy will be greater than 99%. And for example, when you're really working on regions which are repeat rich and regions which are kind of difficult to call regions because homopolymer rich and things like that. So a super accurate model will be much nicer, and it will give you a slight difference in accuracy. So right now, our accuracy is greater than 99% with the single raw reads. So having said that, and then with nanopore, there are some special advantages with nanopore because you are sequencing native DNA. You have the ability to sequence native DNA and native RNA. And because you can sequence native DNA, for example, you can also register the epigenetic information while you are sequencing the native DNA. And the epigenetic information that I'm talking here is, for example, in the context of human, it is 5-methylcytosine. So the signals for 5-methylcytosine can be recognized, hydroxymethylcytosines can be recognized. And if you use some general context models, then you can also recognize bacterial methylation sites. So, but that needs some additional training. So going into the real-time sequencing plots. So when the Minnow sequencing software is running the sequencing program, it also shows you all these kind of sequencing plots, which helps you decide whether the sequencing is going as expected. For example, the channel here tells you, like the light green boxes here tells you that, a strand is passing through the nanopore, and the dark green tells you that the pore is actually available for sequencing, but no strand is passing through the nanopore. So you see there are 265 which is sequencing, at the moment. And then the graph here tells you how much throughput is getting generated. The green line tells you how much is a pass read, and the red line tells you how much is a failed read. And then the figure C tells you what is the quality score information of the reads that you're getting. Is this the expected level of quality score that for a given model that you chose for base calling? So all this kind of information you can get, and also you can get the read length histogram. Let's say you are sequencing an amplicon, let's say a 16S amplicon, so that should be 1.5 KB. So are you getting a big peak at the 1.5 KB region when you're sequencing a 16S amplicon? So from that, you can understand whether your library preparation went well, etc., while sequencing is still happening. So this is really good because, you know, this is only possible because we can actually translate the raw electrical signal into real time sequencing information while sequencing is still happening. And that enables you to actually see all these QC plots while sequencing is happening. And that gives you this advantage of actually making an informative decision. Sometimes if things go wrong, you can stop sequencing, change things, and then you can start sequencing. So that is the great advantage of that. And not only that, and because you get the base calling information in real time, you can also analyze your data real time and make some informative decisions while sequencing is still happening. So for example, if you have reached enough throughput, you can stop sequencing based on if your endpoint is already reached. And all this is possible because you have real-time sequencing and real-time base calling enabled with Oxford Nanopore technology. Okay, so as I said, you know, and I want to also introduce this new concept called adaptive sampling. A lot of interest is in the customer base for this novel technology that's only unique to Oxford Nanopore, because what adaptive sampling means is that when a DNA or RNA passes through the pore, let's say you want to only target some let's say some microbiome, and you know that many times microbiome comes with human contamination, for example. And then let's say you want to deplete the human genome because you may have some human DNA in your microbiome sample, for example. So you can give the entire human genome as your target to be depleted. So when a DNA passes through the pore, and then the actual raw signal gets converted into a nucleotide sequence, and then this nucleotide sequence is actually mapped against the human reference sequence to see whether it is a human reference sequence or not. And if it is a human reference sequence, this particular sequence is rejected from sequencing. And then if it is not a human reference sequence, the rest of the sequencing is allowed to sequence. So this ability to actually know what is a target and what is not a target, and then informatively deciding to sequence, is what we call as adaptive sampling. How do we do that? Is because we basically do a real-time mapping of the nucleotide sequence to the target reference sequence, which is something that the user provides using an algorithm called Minimap2, which is a very fast and accurate nanopore compatible aligner. So it does this alignment in a fraction of a second, and then it decides whether it is a target or not. And then depending on whether you are using an enrichment analysis or a depletion analysis, it will allow you to either sequence that particular strand or eject that particular strand by reversing the voltage. So this is the concept. So, and then let's go to data format. So as a summary for the first section, this is basically I introduce you the nanopore sequencing software and its abilities. For example, it's not just a sequencing software; it also does real-time base calling and converts the and gives you the nucleotide information. It gives you quality control information. It also gives you the ability to either enrich or deplete a certain target. It can also align against a certain reference and give you a kind of a BAM file, for example, for downstream processing. So there are many things that this Minno software can do for you. So having said that, let's go to the briefing on the data formats, because for bioinformatics, it is good to have an understanding of what kind of data gets generated and how the data looks like and what you can do with the data. Because generally bioinformaticians say for any bioinformatic analysis, right? So it's garbage in and garbage out. So basically, if your data is not good, if your data don't have a good quality to start with, then whatever bioinformatic analysis that you do is not going to give you the result that you're looking for. So it is pivotal to actually see whether you have a good quality control of the data, whether you have enough throughput and things like this. For that, you need to have a better understanding of the data formats in the first place and what each and every piece of, what each and every file is actually giving you. So that understanding helps you to actually decide whether you have good quality data and things like that. So the first file that I want to introduce you is a FAST5 file, and this FAST5 file is the record of the raw electrical signal along with other metadata information. So it has this nucleotide information. It has the raw electrical signal, as I show in the right side figure here. It's the squiggle pattern when the current gets disrupted when a nucleotide sequence passes through the nanopore. This is the kind of the pattern that gets generated, and this is fundamental in nature. And then this pattern gets converted into a nucleotide information. So this information is stored in a hierarchical data format called the FAST5 along with other metadata. For example, sequence is one kind of metadata. Methylation probabilities is other kind of metadata. All these metadata are stored in kind of a directory file folder structures in a large file format called HD5 format, which is a FAST5 format. And as long as you have a FAST5 file stored on your computer, you can always use this FAST5 format even in the future. For example, if you introduce a much improved base caller, you can still re-base call the FAST5 files into FASTQ files in the future. So it is always advisable to at least cold archive the FAST5 files for posterity. And recently, we are introducing another format called POD5, which is an improvement over the FAST5 format, because it's much quicker and easier to actually handle these files in real time, and streaming of intermediate files are much quicker. So we are going to introduce this POD5 format and give you an option of choosing between FAST5 and POD5 in the future in the Mino releases to come. Okay, and then the most important file for biologists is the FASTQ file. So FASTQ file actually has the sequence information that you're looking for. So for every FASTQ file has 4 lines for one sequence read. So here I explain you the 4 lines for the one single read. The first line is the descriptor of the read. So for example, the first line will have a unique read ID, and it will have a run ID, and the run ID is common across all the reads in a single experimental run. It will have a sample ID that is user provided, and it tells you the channel number in which the actual read passed through and some other metadata like the timestamp and etc. Second line actually gives you the nucleotide sequence. And then the third line actually gives you the quality score information for each and every base in the nucleotide sequence. For example, let's say if this is a G, and then you have this percentage symbol, which is the actual quality score information written in an ASCII code character. Because ASCII code characters are really nice for computational translations, and that is and it is a single letter code for every single base, and that is the format that is used to represent a Phred scaled quality score. For example, if you take this example G, and G has the quality score represented in the ASCII character as plus symbol. Plus symbol actually means a Q score of 10. That means the probability of this sequence, the confidence level of this sequence is like 90% accurate. So that's what it means for that single base. But whenever we say a raw read accuracy, we actually sum up all the base call quality scores of all the bases in the entire read, and the average of that is what we call as a single read accuracy. With the recent base calling models like the super accurate base calling model, the raw read accuracy of the which is the average of the base call quality scores of the entire the sum of all the bases in a given single read is greater than 99% now, which is a really great improvement. And for example, if you use our latest ligation sequencing kits, and if you do duplex base calling, you can even get 99.8% accuracy these days. Okay, and then remember, these are the 2 important files. FAST5 is your raw signal data file, which also has other metadata, and then FASTQ is your nucleotide sequence file. Basically, every FASTQ file for every single read has 4 lines, and then we generally, as a convention, put 4,000 reads in one file. So that means because you have 4 lines for one single read, you will have 16,000 lines per FASTQ file. So once you get that 16,000 lines, one single FASTQ file gets written to the disk, and then the next FASTQ file gets in preparation, and then when you get another 4,000 reads, you get the second FASTQ file. But many bioinformaticians, people want to know like how much storage space is necessary. So for example, if you are generating 1 gigabase of sequence information, that is in terms of storage, in terms of gigabytes, it generates around 6 gigabytes of storage for FAST5 to actually record the raw electrical signal, and then around 0.5 gigabytes for the FASTQ. So that's with the latest VPZ compression. So this actually, maybe conservatively speaking, you can even call that as 10. For example, if you have 10 gigabase, then you need 100 GB. Of storage space if you have a 10 gigabase of throughput. So I mean, I'm talking conservative, but it is not really 10 gigabase, it's just 6.5. So that is how much space that you need for a 10 gigabase run. For example, when you do bacterial genome sequencing with a rapid kit, the throughput is somewhere around 6 GB, and if you use the latest kits, you can get up to 10 GB of throughput. That means you will need around 100 gigabytes of storage space for a single experiment. Okay. And then some other files that are of like importance is that the sequencing summary text file that gets generated, and this file is generated for every single nanopore sequencing run that you do. So, but basically what this file has for every single read. This the example here is a single read. And then for every single read, it gives you what is the read ID. It tells you what is the length of the read. It tells you whether it is a pass read or a failed read. And it also tells you which channel through which the read passed through and how many seconds it took for the read to pass through the channel. So it gives you a lot of information that can be extracted to run some quality control plots. So that is, for example, there is we have the quality control tutorial softwares in every cloud service platform that we give with Epitome Agent. We have quality control, and that can be done. And then there are also third-party tools like Nanoplot. So if you simply upload the sequence summary.txt file, it can give you nice histograms of your read lengths, read throughputs, all these kinds of files. So this sequencing summary file is something that gives you all the essential information about each and every read. So, but this file, I have to tell you that this file is a big file. If you have, let's say, 10 million reads, you will have 10 million rows representing each and every read in this file. So this file sometimes can be even 1 GB. So this is, even though it's a text file, it's a large file. It needs to be extracted from a terminal. So you cannot just open that from a notepad in a Windows operating system. Okay, and other important files that you need to remember is that whenever we say that you need to upload a reference, the reference file is always, as a convention, is in the FASTA format. FASTA format is basically a text format with an extension called .fasta. Basically, it will have a header line that starts with a greater than symbol, and then it will have a descriptor of whatever it's the sequence. Where, for example, let's say human chromosome 1, the header line will have a greater than symbol, and then it will say human chromosome 1 and something. And then the next line will actually give you the sequence information. Sometimes you can have multiple lines, and every line can have 60 characters of sequence, and then you can have multiple lines separated by a new line character. So that is the FASTA format. That's just a general convention. It is not just a nanopore. It is just a universal bioinformatic convention. Whenever you have sequence information, that is a FASTA file. So when we ask you to upload a reference, that is basically a FASTA file that you are uploading to the software. And then there is another file format called bed file. So what is a bed file is that, bed file actually gives you 3 essential columns of information. So one is, for example, for let's say you have 100 genes, and then these 100 genes, you want to only selectively sequence 100 genes, and then where exactly the gene coordinates of these 100 genes. So that can be registered in a file called bed file, wherein let's say it has 3 columns. The column number 1 will tell you whether it's which chromosome, for example, or if it is a bacterial genome, it will tell you the genome accession number. And then column 2 will tell you the start coordinate of gene, for example, and column 3 will tell you the n coordinate of gene. So row 1 will be gene 1, gene 2, gene 3, like that. So with that, the software can understand, and if you also provide a reference, the software can understand where exactly is gene 1 located, where exactly is gene 2 located, and then it will match your sequence against the reference sequence. And if it falls on the coordinates that you have given for a particular target, and it will selectively either deplete or enrich that particular gene. So for this kind of purpose, you have to feed the bed format file. Okay, so these are all the different format files that you needed to know. I mean, just to have a kickstart on the bioinformatics when it comes to nanopore or any other sequencing platform. FASTA files is very specific for nanopore, but FASTQ files are very generic. It just the convention is the same across Illumina or nanopore. And BED file, the convention is the same. It is the universal genomic convention, and then basically it has the chromosome information, the start coordinate and the end coordinate, and the CSV file, the sequencing summary.txt file, is a very unique thing for nanopore. It's basically a plot that actually registers each and every read that comes out of the nanopore sequencing exercise, and then based on that you can actually do some quality control. Okay, so that's a recap of the file formats, and let's go into bioinformatic basics now. So when you start any bioinformatic exercise, right? So how we would like to put that is that, it is not a one-way street, in the sense that, let's say if your project aim is to do a bacterial genome assembly, if that is your project aim, you cannot first generate the library preparation and generate the data and then think about how I can approach a bacterial genome assembly. You have to do the planning even before starting the library preparation. Why is that? When you want to have a certain answer, the data has some requirements. For example, if you are doing a bacterial genome assembly, you need to have a certain amount of throughput. Let's say an E. coli genome. A E. coli genome has a 5 megabase genome. For example, an average E. coli genome is 5 megabase. And let's say you want some 100x coverage of the 5 megabase genome. Because an assembly project, if you use a certain assembler, it requires 100x coverage to give a nice and decent assembly. So what is 100x for a single bacterial genome? 5 MB into 100 is 500 MB. So for a single bacterial genome, you need 500 megabase of data. So if you have 10 bacterial genomes, that means 500 MB into 10, which is 5 gigabase of genomic data you need. So if you need 5 gigabase of genomic data for 10 samples, that means you can only multiplex 10 samples, and you need to know the throughput that you will get from the kit that you're using, whether this kit can give you 5 gigabase of throughput. With that, you can decide whether you can multiplex 10 samples or not, and will it give you that much coverage that you need. So all this planning needs to be done dependent on what is the downstream analysis requirement. The data directs the way in which the experiment has to be planned and done. So that is why we call that it's always a two-way street. So you need to actually first start what is the objective that you want, and accordingly you have to choose a different library preparation kit. Another example that I want to tell you in the context of bacterial genome sequencing is that people want to also sequence plasmids when it comes to bacteria. But plasmids are circular in nature, and if you use our conventional ligation sequencing kit, it is difficult to actually cut these plasmids out. So we have this rapid sequencing platform, which are transposase based, which can randomly cut genomic DNA at different places, and you can tagment the adapters for sequencing. So if you want to also get the genome assembly of plasmids, it's good to go for the rapid sequencing kit because it's transposase based. You can cut the circular plasmids and linearize and put the sequencing adapters so that sequencing can actually be done. So based on if you want a plasmid genome assembly, if that is your objective, then you need to know that you have to choose the rapid kit for library preparation. So this is the idea behind thinking, I mean, from the end to the top to actually know whether you have a right decision for a given experiment. And then, as bioinformaticians, you need to actually know all these file formats, and that's why I gave you an introduction for file formats even before starting any bioinformatic tutorial. You see, we have covered many of these formats, some we haven't covered. This is results files. For example, if you do variant calling, then it is generally recorded as a variant call format file. Basically, what is a variant is any base that is related, it's different from the reference. If you take a read and match against the reference, and then you have a nucleotide sequence in the read that is different from the reference, then that is what you call as a variant. But it is not decided by a single read, it is decided by a pileup of reads, and then a consensus is calculated, and then a variant is called relative to the reference. And these variants are stored in a file called variant call format file. And how the file should look like, what will be column 1 in the file, what will be column 2 in the file, what will be column 3 in the file. If you know that, then it is good for you to extract a certain piece of information as a bioinformatician. Okay, so these are all the basics about file formats that you need to know if you want to do any bioinformatics. Okay, so let's give you a small introduction about command line. You know, I assume that many of you are biologists and there are some bioinformaticians. So I'm a PhD in microbiology. So I mean, I'm a bioinformatician by practice over 20 years. So I know the trouble of a biologist in actually when it comes to a terminal-based analysis. So as a starter, what I would tell you is that for people who want to learn bioinformatics, you need to at least learn a little bit of how to actually use command line, because for to be honest, I mean, if you want to grow yourself as a bioinformatician, for most softwares, it is compatible with the Linux environment. It is more compatible with the Linux environment. Not that the Windows environment cannot do it, it can do it, but sometimes you see all large servers are Linux based, right? So I mean, Linux workstations, you can actually scale them up at the server level, and you can do a lot of analysis. Most of the bioinformatics softwares are written thinking that the bioinformatician is going to use a Linux environment. And even in Windows these days, you have PowerShell, for example, where you can write commands. So it is good to learn a little bit of commands. Like, you know, I'll show you here. There is a command sheet that you can start with, and that will tell you like small commands, which will tell you like how to actually change from one directory to another directory. For example, CD. If you type CD and then one directory, it will change you to that particular directory. LS means list. You could list all the contents in the directory, just like how you browse and navigate to a folder. And then, for example, PWD is print working directory. For example, if you don't know where you are sitting, then you can just do PWD. It will tell you which is the folder that you are sitting. CP means copy, for example, copy one particular folder or file to a different location. MKDIR means make directory. You can create a directory, like how you create a directory using a browser, like right click and create new folder. You can simply type MKDIR and folder name. That will give you a directory through the command line. MV is moving one and renaming at the same time one file or folder to another file or folder. RM is a very dangerous command, which is called a remove. In a Linux platform, if you type RM and then remove a file, it is removed. There is no recycle pin. So it's very hard to when you remove something. So you have to take care when you try to remove files in a Linux environment. wget, for example, is to try to download something from like, you know, in HTML, like, you know, anything that is available on the web, you can use wget and give the URL. You can download things. Man is manual, for example. Anything you want to know about any command, you can type man and that command, and it'll give you some kind of information about what that command does for you. So you don't need to remember and things like that. So you can practice all these things on your terminal, either it's a Windows PowerShell or a Linux environment, whatever you have. You can practice some commands. Some of the commands will have some slight differences relative to whether it's a Windows or a Unix operating system, or if it is a Mac OS. So the commands will be slightly different, but the concept is the same. So everything is a single line of command. You can practice and get your hands on within a week, I would say. And then in the context of nanopore sequencing, how this is helpful is because often you can do many single line of command execution to actually run a certain process. For example, if you want to do offline base calling, let's say you have the raw electrical signal data generated with a nanopore sequencing experiment, and you have the FAST5 files, and you did not actually base call them into a FASTQ file while you are sequencing, because let's say your computer didn't have enough firepower to do sequencing. Like Rifki pointed out that, you know, you transfer data to your data analysis server, like your DGX workstation, and then later you want to do the base calling exercise in your DGX workstation. How do you do that? So you can run the standalone command, for example, from the terminal. So here is an example of how you do base calling from a terminal. It's a single line of command. So basically that guppy base caller is calling for that particular program. The program name is guppy underscore base caller. And then you just need to give this dash dash means a parameter. Flow cell is a parameter. And then this flow min 106 is a value. So you have a parameter and a value. So what are all the parameters you have? You have a flow cell parameter and you have a kit parameter, and then you have I dash I is an input parameter. You have to tell where the file is. And then dash S is output parameter, like, if you base called the FASTQ files and where do you want to put them. So that is the out parameter. So as you can see, it's a single line of command, which tells you, it calls the base caller program, which is guppy base caller, and it tells what is the flow cell that you used, and it tells what is the kit that you used, and then it tells you where is your file located, the FASTQ files, the raw data, and where the FASTQ file should go. So it is as simple as running this single line of command is what you call as just some terminal based bioinformatic exercise. So as you can see that it's simple, that you can, any biologist can learn, and that is why we say, like, sequence anything anywhere by anyone, because this is something that anyone can learn. You just, if you take step by step, you will certainly get there. And then you see, you don't need to remember all these commands for every program, right? The convention, when software developers write code, right? The convention is for every single program, there will be a help. So if you do this dash dash help, for example, if you type in the terminal guppy underscore base caller and do dash dash help, it is going to give you all the information and all the different input parameters that can be given. For example, you can have a parameter like scaling, trim strategy, so many parameters, and it also tells you what are all the what that particular parameter does. And then it also gives you on a simple level, what is the simplest possible form of a command. So the simplest possible form of a command where the software requires you to supply some mandatory information is your input data and your output path, and then the flow cell information or the kit information. Alternatively, you can directly give a config file, which actually summarizes the compatibility for flow cells and kit. So this tells you, like, what is an example command, what is the essential information that you need to type and hit enter, and then it will do the job for you. And then all the data that you want will be stored in the location that you actually specified. So, and here I show you an example for demultiplexing your data on a file server, for example, your DGX workstation. You transferred your data and you did a base calling exercise by running the command that I showed you in the previous window. Now you have FASTQ files. Now you want to demultiplex the FASTQ files. Let's say you have like 10 barcodes and you want to demultiplex them, and then you want to say barcode 1, barcode 2, barcode 3, all reads from barcode 1 should go to barcode 1 and barcode 2 and barcode 3, so on. So you run this single line of command which says guppy barcoder, which is another program in the suite of programs that comes with the guppy software. So you are calling the specific guppy barcoder program, which does the demultiplexing for you. You give the input path. In this case, this is the FASTQ file, and then you give a save path, and then you give what kit did you use, and if you want to trim the barcode sequence, and you want to give a score, and then you want to have barcodes at both ends. In the case of SARS-CoV-2, for example, if you're using a classic protocol, things like that, you can actually specify some optional parameters. And finally, the end result of this is that you will get all the reads like pinned according to the barcodes. And later you can take a single barcode for downstream analysis, or all the barcodes together if you are using our platform like Epitome, etc. So as you can see that, bioinformatics is not that difficult. Most often it's just running single lines of command for a starter, and it can already do a lot of things for you. Okay, so having said that, and I will now introduce you to some ONT bioinformatics solutions that we have, because we strongly, our vision is such that, we know the biologist pain, and we don't want to be just a data generation sequencing company. We also want to enable people to actually analyze their data, because more often the problem is that when you do like, let's say, other technologies, you have to rely on some bioinformatician, you have to rely on some high throughput data center. You have to collaborate with some bioinformatics lab, and then you send your data to them, and they have to do your analysis. So that actually takes time, and has some communication lost in translation and things like this, if a biologist can analyze their own data, that is the best deal that can happen. So at least, I mean, even if you cannot analyze the data to their entirety, at least you should know the basics of data analysis, so that, you know, when you communicate with the bioinformatician, then you know what you're talking, and that way you can get the results that you want. And even if there is some mistake, because the bioinformatician on the other end may not know the biological nitty-gritties of the data and the sanity check from a biologist point of view, he cannot do it because he didn't understand the data as a biologist understands the data. So that way, biologists knowing a little bit of data analysis themselves really go a long way. And that's why ONT really tries to generate a lot of free software tools that biologists can actually learn and use, and also learn to interpret the data. Okay, so what are all the solutions we have? So we we split our solutions into 3 larger groups, and the one is a cloud-based informatics solution. So the cloud-based informatics solution needs a software called Epitome Agent. So this Epitome Agent is just a data uploader software. What it does, when you do sequencing, your data gets generated into, let's say, your sequencing device like a grid ion or a min ion. If it is a min ion, then the data gets generated onto your laptop or your workstation. And if it is a Promethean, data gets generated into the Promethean server. I mean the Promethean data acquisition unit. And then if you have internet connection, and if you don't have time, or if you don't have the expertise to do any bioinformatics, you can simply upload the base called FASTQ data into our cloud server. And we have hosted a lot of numerous data analysis servers on AWS, and we will do the analysis for you. So this Epitome Agent software is something that you need to download onto your local machine. And then this software, all that it does is that it will help you to pick a pipeline. And then let's say if you want to do 16S data analysis, it will help you to pick the 16S data analysis pipeline. It will ask you to tell where the input folder is, and it will ask you to actually fill where the, other small parameters for that is specific for a particular analysis, and you can click start. When you click start, data is going to get uploaded. And then the results are going to get automatically, some of the results will automatically go to the designated output folder, and some results you may have to download from the web. So that is the point and click solution that we have for biologists who don't have time, or clinicians who don't have time, or you want to kind of get a quick analysis. Let's say in the evening you want to just simply upload the data and go home, and then the next day morning you can already have the results on your desktop. And this solution became really handy for people who did SARS-CoV-2 analysis, because many clinicians, for example, they do not have the time to learn the bioinformatics. All that they do is they choose the SARS-CoV-2 end-to-end pipeline from Epitome and simply upload the data. Next day morning, they already know whether it's an Omicron or a Delta variant or whatever. So that is as simple as that. And then we have the next solution, which is a bit bioinformatics savvy, which is called the Epitome Labs workflows. This is based on a workflow manager called Nextflow. And basically, this helps you to actually efficiently resource all the compute that you have in your machine. So it is good for actually a server architecture, for example, or you can run in a laptop. You can accordingly use the CPU and the GPU or whatever memory resource that you have. It's very scalable, both from a desktop standpoint to a workstation standpoint, or even to a server architecture. Very scalable and production level workflows. The same workflows that is there in the Epitome Agent is actually there in the Epitome Labs. But sometimes some workflows that we have in Epitome Agent we don't have. But the pipeline, when we register a pipeline, the pipeline, for example, if you are using a human variation pipeline, the pipeline that we have in Epitome Agent and the Epitome Labs are the same. It's just that if you don't have time, you can upload data and we'll do the analysis for you, or you can do the analysis on your own using the Epitome Labs. And Epitome Labs can be sometimes faster, because if you have a high performance server, then you can run the analysis much faster than the cloud can do. Because sometimes the cloud is supposed to be real time, but on the other hand, we cannot control how many users are using a particular cloud service. Sometimes you can get the results in 20 minutes. Sometimes it may happen that you can get results in 1 day. So that way, if you want to have time control on whatever you are doing, you can do the Epitome Labs Nextflow based workflows. But for this, as I said, you need to learn a little bit of command line and things like this, which will come in handy. So the last one is for a kind of a seasoned bioinformaticians, which is our research software releases, which you can get from Nanopore GitHub. And we have like more than 80 packages there, which are research releases where you can actually train your data for a certain experiment. And then, our research level base colors, like, new base colors, Dorado and things like this, and our methylation color, and many research level tools are available on the GitHub for Nanopore. And if you use these research softwares, these are purely Linux based. So you have to download that into a Linux environment, and then you have to install it, read the README file, and then you have to do your data analysis. For this one, you need a little bit of a bioinformatic experience. And when you encounter some issues, you can directly contact our developers who are available for you on the GitHub repository page. You can post an issue there, and they will directly contact you. Okay, but for general purpose, we would recommend for most people the Epitome lab workflows and then the Epitome agent workflows, depending on your level of expertise. Okay, and then why we need Epitome for data analysis? I mean, when I talk about Epitome Cloud, what is so special about Epitome Cloud is that it can do real-time data analysis. I mean, the definition of real-time here is that while sequencing is actually happening, you are uploading your data and your data is getting analyzed while sequencing is still happening. So this Epitome agent software that you're installing on your laptop, right? This software is going to constantly look into your input folder for new data. So when it finds new FASTQ files, it's going to upload the new FASTQ files. When it finds the first 10 FASTQ files, it will upload the first 10. And then after that, the sequencing is going to generate another 10 FASTQ files. It will upload the next 10 FASTQ files. And as and when it uploads more and more data, the analysis will keep repeating, and then the analysis results will keep updated. How this is useful, let's say you are doing some metagenomic experiment, and then you want to know, let's say, a pathogen contamination in your water sample or something. Let's say you're looking for Shigella contamination, and then you are running a hospital sample, some taken from swabs taken from somewhere, and then you want to look for whether there is Shigella contamination or not. While you are sequencing, if you're uploading the data, you can already see the classification tree, and you can see whether you have Shigella or not, or Bordetella, or whatever you want to see. And if you already see that, if you see that in enough coverage, you can already stop the run based on the analysis result that Epitome agent is showing you. So that is why we call this real time. Even though this is not instant, this is happening while sequencing is still happening, and we want to call that as a real time analysis. Okay. So, and then how it works, I mean, it just you upload data and then we do the analysis for you. You see, this is an example of a taxonomy tree that comes from a program called What's in My Pot, which actually does the metagenomic classification for you. And as the sequencing is happening, more and more data gets generated. This taxonomy tree gets updated and it becomes more and more denser. And then let's say you are looking for Shigella, and if you already find Shigella, and then it also tells you how many Shigella reads are there in the entire population of reads. And then let's say if it's a predominant contamination or things like this, all these kind of information you will get real time. And when you reach enough density, you can already stop the run if you already got the answer that you're looking for. So that is the kind of use case for actually using these kind of real time analysis. Okay, so and the other thing is, it just point and click. You just simply upload your input data. I will later show you how to do it, and then you just choose the whichever workflow that you want. We have several workflows on Epitome agent. You choose the workflow, click the workflow, and then just fill in some basic parameters and start the workflow. It will upload your FASTQ files and it will give you results. Okay, and then the Epitome workflows. This is also, we have another software called Epitome Labs that you can download from a website called labs.epitome.io. And this software that you download locally on your computer, it will give you this Nextflow based pipelines. And you also need another software called Docker, because all these pipelines are kind of containerized into a Docker image. So basically, you can imagine Docker image as it's a software that has its own internal environment, unconnected to your outside environment. So there will be no conflicts, and anybody can do Dockerized data analysis, irrespective of whether you are in a Linux environment or a Windows environment or a Mac OS environment. So it gives you reproducibility, and then it solves you a lot of problems from incompatibility because of softwares coming from your inbuilt operating system conflicting with some of the installations that you did. So Docker Dockerization is a really a modular use case for actually running software analysis. So the entire software goes as an image inside a virtual container. So you can imagine that as its own operating system with all the softwares necessary. So all the Epitome labs run inside, every workflow runs inside a Dockerized container. All these are pre-packaged in the Epitome lab software for you. So once you install Epitome labs, all this comes pre-packaged for you. You can choose a workflow. It gives a graphic user interface. You can click a workflow and it will show you a form to fill the basic parameters, and then you can click launch, and then it will launch the next flow workflow for you at the back end. And then it will give you a HTML report file. And if you're running variation analysis, it will give you specific files like VCF files. And if you're running alignment, it will give you BAM files and things like this. Okay, so it is very scalable. Why do we have these flavors? Because the Nextflow based pipelines are production workflows. They are very scalable, and that can suit a server environment. Okay, so and then we have several pipelines, the Nextflow workflow pipelines that comes under the Epitome labs. So for example, WF human variation, single cell transcriptomics, we have a bacterial genome assembly pipeline, and we have a bacterial clone validation pipeline for plasmid assembly. And so these are things that only Epitome labs has, because these are compute intensive. So we cannot do the bacterial genome assembly, or this kind of bigger projects which involve a bit more compute power. So the bacterial genome assembly, for example, is only available as a Nextflow pipeline, for which you need to download the Epitome lab software. Okay, and then like I said, we have the open access, the GitHub research releases. There are like 80 research releases you have. For this, you have to be a little bit more bioinformatician, or you have to learn to become one. Okay, and then we also don't restrict our customers not just for our software. So you always find all application specific tools on our resource center webpage. Anybody who has a really nice software for any nanopore data analysis, for example, in the case of 16S, right, there is a very popular software called NanoClust, which is a third-party software, but the NanoClust is better than the 16S pipeline that we give in ONT, because the ONT 16S pipeline will do a read-based analysis. Every single read will be classified against a database, but NanoClust will take all those reads, bin them, polish them, make them more accurate, and will take a representative read and blast against the database, and that gives you a species level accuracy. So people who do day-to-day 16S analysis actually choose NanoClust. So how to know whether which kind of softwares exist? Then you can go to our resource center. We constantly update new publications on the resource center. So any application you type bacterial genome assembly, if there is any third-party pipeline that suited for nanopore long read has come, we will publish that in the resource center. You can take the things. So we have our own pipelines. We have third-party pipelines wrapped into our pipelines, and we also have a large user base and the community base which constantly develop nanopore softwares that you can get from our resource center. Okay, so with that, I will stop the theory part, and if you have any questions on the theory part, you can ask. Otherwise, I will, if you want a small break like 2 minutes or 5 minutes, you can do that. Otherwise, I will go into the terminal. I will show you practically how to launch some workflows, both on the terminal and also using some Epitome agent analysis. So Rifki, so what shall we do? Do you want a 3 minutes, 5 minutes break, or we can directly start practicals?
I think I will ask the audience whether some questions from floor.
Okay.
Yes, open mic. Raise your hand. I think there is no questions.
Okay.
But I think I would like to have I would like to have one question about Epitome.
Okay.
Is it possible that we have Epitome on computers, like
you are breaking a little bit, Rifki, so I cannot hear you. You are breaking a little bit, so can you repeat me again?
Okay. At BRIN, we have HPC clusters.
Okay.
And is it possible to have Epitome systems installed on premise so people can?
Yes. I mean, if your clusters have a let's say Ubuntu operating system with a desktop interface, you can install, and then you can use the desktop graphic user interface to upload data. But if it's only a terminal interface, there are client programs which can do the same thing that the graphic interface can do. So, but you need an internet connection for that.
Yeah.
Yes.
Of course. So do you have experience or Nanopause has experience with the like other institute where the analysis platform are maybe centralized in the HPC system?
Well, I don't know in the sense like, I mean, if people have, like in HPC systems, if people are running, and I'm sure that so many people are running and centralizing things, but I don't have any specific use case in my mind, but technically this is possible.
Okay.
Yeah, I mean, the more because you have a HPC cluster, the much easier one is a command line interface where you can just batch analyze a lot of data. I mean, we have this Nextflow based Epitome Lab workflows, right? So these workflows are actually like, designed to be scalable to work in a HPC cluster. So many people are running that. So for example, human variation analysis, right? People don't want to upload gigabases of data. So you can run the WF human variation workflow on your HPC cluster with you can maximize the compute resource. You can tell how much RAM you need, how much CPU you need for a given pipeline, and you can control the pipeline and monitor the same. And you can also batch process the data.
Yeah. Is there any question from audience? If not, we can go on with the.
Okay, so let me do some data analysis for you because you are interested more on bacterial genome assembly. So I will show you practically how do I launch a Nextflow based bacterial genome assembly pipeline from a terminal. Okay, so this is an exercise that I want to show you. And then while that exercise is running, I will also try to show you how the Epitome agent cloud-based point and click software works. So both we can do, and let's see if we generate some real-time data before this session finishes. And if you're lucky, we can do that. Otherwise, I have already analyzed some test data set and I'll show you how the output looks like. Okay, so let me do some share screen. Okay, do you all see my terminal?
Yes.
Okay, perfect. Okay, so this is a Promethean, which I have connected myself to a Promethean remote server. So this is one of our Oxford Nanopore devices that we supply, which can do the large level human sequencing. I mean, these Promethean flow cells have it's so big, it has like a 12,000 pores, and then it can do, it can generate 150 gigabase of throughput for an experiment. It can even generate 200 gigabase of throughput for an experiment. So you can get 30 to 60x of a coverage for a human genome from a single Promethean flow cell if you optimize it nicely. So that is the server that I have connected my Windows computer to, and then I'm already logged into the server. So this Promethean servers has a data folder, which is basically like, it has like if it is a Promethean P24, it has like close to like 30 terabytes of space, and then we have this data folder in which you do all your experiments and store all your data. So I am inside that one such data folder, and I have a folder called software. I hope you can see what I'm typing. Let me go to the top of the thing. So here I'm typing ls. ls means list. So I want to see what is inside this software folder. So, okay, so I see 4 things inside the software folder. There is an analysis folder that I've created, and then there is some other folders that I've created. I have downloaded already the WF bacterial genomes folder from GitHub. For example, what is GitHub? So let me show you. So here. So this is our Nanopore repository for GitHub. I hope you are seeing my web page for GitHub. So we have several softwares that you can get from our Epitome labs, for example, WF transcriptome, single cell, and many things. You see the WF bacterial genomes pipeline in GitHub. So if you click this pipeline, you see that, you see this thing called code. You go here and then you see this particular thing. So this is what you need to actually copy and then download this particular folder to your computer. So that is one way of actually taking the entire folder. And then this is what I did already. And then you have this folder called WF bacterial genome sitting here. Because what I do is like, I say git clone, and then I put that URL, the web URL for the WF bacterial genomes. And if I type the command git clone, Git is a software that can download for you. And when I type that command, the whole software will be downloaded and it will be named as WF bacterial genomes. Okay, so that is one pipeline for bacterial genome assembly. I'm showing you this next flow based pipeline on a terminal. So I want to go inside that folder. So I use the command cd. Cd means change directory. And then I want to go inside the WF bacterial genome folder because I'm in a one level away from the WF bacterial genome folder. I'm in the software folder inside which the WF bacterial genome folder is there. So I want to go inside that. So let me go inside the bacterial genome folder. So what I did that you cannot see now is that I just only typed WF and then hit the tab button. In Linux, the tab button can auto-fill the remaining for you as long as it is that particular name is non-redundant. Here, there are only 4 folders and only one folder starts with the prefix WF. So when I simply type WF and hit the tab key, it will auto-fill for me the entire name. Okay, so now I hit enter. I'm inside the WF bacterial genomes folder. I will type something called LSLRT. So LS is list and LRT will also give me timestamp of when and each and every file is downloaded. So it gives me in this kind of a list fashion, like all the different contents which are present inside the WF bacterial genomes folder that I downloaded from GitHub. And here you can see there is a test data folder. And then for every program you download, every software you download, the convention is there will be always a README folder. I mean, README file. So you can always go inside the README file and learn about what that software program is doing. So one way of going inside the file is a VI editor. So I type VI, which is an editor, which is another program which is helping me to open that file called readme.md. So I'm going to open that file, and then this is the information inside that file. So what is it saying for you? It is telling you the workflow can be currently run using a Docker container or a Singularity container. And it tells you if you want to know more about the workflow, because it's a Nextflow based workflow. So you have to type the command nextflow and then say run, and then it is an Epitome Labs workflow. So you have to type Epitome Labs, calling the Epitome Labs program, and then inside which you have the WF bacterial genomes pipeline. That is one pipeline among the list of pipelines that are inside the Epitome Labs. So you type the WF bacterial genomes pipeline and then you type -- help. And then it will give you a list of parameters and which will tell you what are all the different things that you need to supply. For example, what is the input file you need to supply? What is the output file you need to supply? You can get to know from the README file. And then it also tells you, if you go down the file, it tells you, okay, what are all the kind of file formats you will get? What is the output file you will get? What is the kind of input files you need? And what programs are used for this analysis? All these are inside the README file. Yeah. So I now type Shift:Q to quit this file and come out to the terminal window. Shift:Q. Okay, Q for quit. Okay, so I quit the README file. Now I understand a little bit about the program and how to use the program. Okay, and then every program will come with a test data because you might have not generated this data. I mean, but you want to actually run this pipeline with some test data. So how do you get some test data? You can already get the test data when you download the package from GitHub. So I CD into the test data folder. CD means change directory, and then I list the entire file contents. I go inside, I see a FASTQ folder. So FASTQ folder is the nucleotide information that I was talking about. I change directory into the FASTQ folder now. Okay, because I want to see what is inside the folder. Okay, now I see there is a barcode1 folder, barcode2 folder, and there is a subset.fastq.gz file, which is basically for ease of purpose. I have subsetted some data and then put it in the outside level. But let's go into a barcode1 folder. What do you have? You have this file here. And I want to, let's say I want to see I'm some contents of this file, let's say, let me just move this panel a little bit here. Okay, so let's say I want to see some contents of this FASTQ file. What would I do? There is a command called zcat. Okay, so I say zcat, and then I type subset, which is the file, and then I can, in Linux, you can use this operator. We call that as a pipe operator, which will actually output the, the analysis of the previous program into the next program in succession. So pipe, and then I want to just see some lines. So let me say, I just want to see some 10 lines. Okay, it shows me, okay, this is the nucleotide sequence, so which is inside the FASTQ file. Okay, so if I just say head -1, so it'll show me the header line. Okay, so the header line, that thing that starts with @ symbol, is the unique read ID. Okay, so this way you can know what is inside the FASTQ file. If you want to have some sanity check of the FASTQ file, this is what you can do. So if I do head -2, then it will give me the second line is the sequence. You can also do tail -1, which will give me the last line. Tail -50 will give me the last 50 lines and things like this. Okay. So I go out and I clear the window. Okay, so I come back here. So I saw some FASTQ file. I just did some sanity check with the FASTQ file. I know the file has sequencing data information and things like this. So now let's go and run some analysis. Okay, so cd // means go one level, the one previous level up. So if I do cd // it will take me to test data. If I do another /, it will take me to bacterial genomes level. If I do another /, it will take me to the software level. So I'm trying to go 3 levels back. Okay, and now I am at the software level already. Now I list. So these are all the things that I have. Okay, so I already made an analysis folder. Let me go into the analysis folder. Okay, and I'm typing what is the contents of the analysis folder again. So what I do generally is that I create a file called shell file. This file is kind of, you can imagine this file as a file where you can actually type your commands and save it. So that, you know, whatever you typed on the terminal, you saved it as a kind of a notepad file, so that you can see later if you made any mistakes or anything, you know, you can also use this file as a log file. At the same time, you can use also that file to run the command that you want. So let's see what I have inside that file. So I can use the command cat to actually print the contents from that file. So I already made that file. So I am printing the contents from that file using the cat command. So basically typing cat with a space and then the content, the name of the file, and hit enter. If I do that, this is one single line of command inside that particular file, which says nextflow run. Basically, it is calling the program nextflow, and it is asking that nextflow program to run the Epitome Labs workflow. What is the name of the workflow? WF bacterial genomes. And then this symbol is basically a line separator. Okay, and then it gives you one single parameter. The parameter is the FASTQ file, which is your input file. So where is that file located? This is the value of the location of the file. So that is located in /data softwares. Inside that, the WF bacterial genomes folder. And you remember the test folder that we downloaded. And inside that, you have a FASTQ folder and this particular file. So as simple as calling the program name and then telling what is the pipeline that you want to run. And then you simply give the file name and it will do the analysis for you. I have typed the entire command inside this file called runbackgenomes.sh, and now I'm going to, let's say, execute that one. So okay, so I see this is a shell file, so I execute using the command sh. So if I hit enter, it is going to run the workflow for me. It's as simple as executing a single line of command to run the entire bacterial genomics workflow pipeline. You see, these are all the individual steps that pipeline is running, because this WF bacterial genomics pipeline actually runs through several different programs. For example, you have the reads. This is an assembly project, right? So you have reads. So these reads, based on an overlap layout consensus algorithm, the reads will be just aligned against one another, and then like the small threads will be aligned against one another like a zigzag puzzle, and you will get a larger thread which we call as a config. Basically aligning and overlapping all the reads on top of each other and trying to build a large single thread. So that we call as it's a larger piece of the nucleotide information, like a jigsaw puzzle aligned together from end to end, and then you get something called what we call as a draft assembly. So you need some aligner for aligning the reads. And then after aligning the reads, there are several reads in each and every location, right? For every genomic piece, there are several reads. So you need to take some consensus from each and every read, because a single read may have an error, but another read for the same position may not have the same error at the same position. So when you have 100 reads in a given area, 99% of the reads will actually have the true base. So maybe one read has an error, but 99 reads for that given position will have the correct base. And with that, you can do polishing because you have a consensus, because you know what is the majority in a given position. So with that, you can do polishing, and then you can find out exactly what is that particular sequence. So this software does read alignment, and then it does contig building, which is basically aligning all those small pieces into big pieces. And then finally, it uses our in-house software called Medaka to actually do further polishing of the entire contig. I mean, after we build this draft assembly, we again take the reads and match against that draft assembly sequence that we built, and then further refine each and every base, whether it is correct or not, by actually finding the consensus once again. So this is what we call as polishing, and for that we use our own neural network program called Medaka. And after Medaka does the polishing, it does something, and it takes the program, I mean, it takes the output of the polished assembly into something called PROCA. So PROCA is a bacterial annotation software. It comes with a bacterial backend database, and what it can do is it can find the coding regions, it can find the ORF, it can find the gene annotations, because you basically end up with a genome sequence at the end, right? But you don't know where is gene 1 start point, where is gene 2 start point, end point. You don't know any of these information, right? But without that, just a simple sequence from head to tail doesn't make any sense. So you need to annotate that sequence, and you need to know where is the your, let's say, gyrase A gene is present, where is your rec A gene is present. If you have some antibiotic resistant genes, like BLA resistance gene, then you want to know where is the coordinate of the gene. So this annotation is done using the software called PROCA. And finally, we do make reports. And make reports will generate HTML reports of the entire workflow. And this way you can finally see as a summary file from start to end with some QC files to start with. And then it will give you the crux of the analysis and all the summary files as a HTML file, which you can share with your collaborators. On top of it, you will get an assembly, which is your final output that you're requiring. So you will get an assembly consensus.fasta file and other log files, etc. So currently this pipeline is running. I know I have tested this pipeline, and I know that this pipeline takes around 9 minutes to finish. And while this is happening, we can maybe do a parallel epitome agent run. So this is a terminal based how to launch a bacterial genomics workflow. So while this is running, instead of wasting time, we can do some epitome agent analysis from my Windows laptop. Okay. So the epitome agent, just to recollect for you, epitome agent is a point and click interface to do the data analysis on our cloud. Okay, so all that you need to do is simply upload your FASTQ file. Okay, so I'm going to do the search button, and then I'm going to click the epitome agent software from my Windows laptop. Just remember, the other analysis is running on a remote server. I am connecting my Windows laptop to the remote server, and the black screen that you're seeing behind is actually a remote server, which is a Promethean data acquisition unit, in which this particular Nextflow pipeline bacterial genomics assembly is running. But parallelly, I'm going to start a run of the epitome agent software from my Windows laptop. Okay, so let me click the epitome agent. Do you see my epitome agent pop-up interface? I hope you see.
Not yet. I think you still share the.
Okay, so let me share the entire screen so you can see everything. Okay, instead of sharing just the command line, so I will share the entire screen. Okay, so now do you see? Rifki, do you see now?
Yes.
Okay, so now I have the epitome agent interface. I mean, I double click the epitome agent software, which I have already installed, and this opens, this is the graphical outlook that you see. All that it asks is, it asks me to drag and drop some file for doing the data upload. Okay, so let me open something from my computer. Okay, so I have, so if you don't see something, you please tell me. So I'm opening some file from my computer just to show you. Okay, so let's see what I have. So let me go to some data sets that I can upload. Okay, so I have a data sets folder here. Okay, let's upload some data to the epitome agent. Okay, so let's upload metagenomics data. Okay, so what do I have? So I have a metagenomic sample. I have a FASTQ pass folder. Okay, so you see inside the FASTQ folder, FASTQ pass folder, you have several barcoded folders. So each and every barcode is a single sample. Assume this is as your each and every sample is your metagenome sample or your bacterial sample or whatever. All you need to do is just simply drag and drop the entire FASTQ folder. So I just drag and drop the entire FASTQ folder, and you see that FASTQ folder is located here. It got drag and drop into the epitome agent interface, and I click the next button. And now once I click the next button, let me maximize this. Once I click the next button, now it shows me all the available workflows on the cloud interface. Let's see if I want to do, okay, what I want to do, if I want to run the metagenomic pipeline, which is called what's in my pot, basically what is there in my sample. So that is the pipeline that we have for, metagenomic read classification. So you have to choose this particular pipeline. This number is the version of the pipeline. So that was uploaded on 2021, November 11, 26. Sometimes we will have two versions of the pipeline, one recent pipeline and the version just before the recent pipeline for, if you're writing some publication and if you ran a pipeline with a certain version number, you want to have it. And while you're writing publication, so we at least keep one version and the version before it. So let's choose the FASTQ WIMP pipeline for this analysis by clicking that thing. And then we always ask for the consent for whether it is a human sample or not. Okay, so if it is not a human sample, then you have to say no. And then there's a parameter filling page, which actually asks the user to fill some basic parameters that is specific for a particular pipeline. In the case of metagenomic pipeline, it is asking me what Q score do you want? So Q score is basically the average raw read accuracy of each and every read. Let's say I don't want reads which have a Q score of less than let's say 8. Okay, I say the FAST model gives you a default score of 8. So I want only reads which have a Q score of 8 and above to be uploaded to the database. So I choose a Q score of 8. Okay, and then I say, okay, I don't want reads which are less than 100 base pairs in length, because those reads is not going to give me nice results if I think. Or if I want reads only greater than 500 bases, I can say, okay, I want reads only greater than 500 bases. Okay, that is a minimum length filter that I can give. So let's say I have 1 million reads, and then out of that 1 million reads, 100,000 reads are less than 500 bases in length. Then only 90% of the workflow will be successful, which means 10% of the reads got filtered based on your parameter. You can also give a maximum length parameter. I mean, that is only useful in situations, for example, if you're running a 16S amplicon pipeline, you know the amplicon length is 1.5 KB. So if you have a 3 KB fragment, you know that it cannot be your amplicon or some chimeric DNA or whatever. So you can also give a max length parameter if you know the decisively the fragment length of your amplicon. So in this case, it's a metagenomic sample. It's of different fragment lengths, so I don't choose a max length parameter. And then finally, there is an output folder. You can change your output folder. And especially when you're running on our devices like Gridan devices, Promethean devices, these softwares are present in those devices. If you're running them, I would advise you to actually output the folder to the output the data to the data folder, because data folder has all the space for data. But there is also a home folder which is taking the system volume, which has only 60 GB of space. So if you're running in the home folder, then it may get filled up, and then your analysis may stop. So you have to make sure that your output folder has enough space. And then download mode, it says data and telemetry. So what does telemetry mean? Telemetry means that when you upload each and every read, right? So it gives you some log information registered on your server, which tells you whether the read is successfully uploaded and what it is doing. Is there some preliminary analysis is done? So there is a verbose log if you actually choose data and telemetry. And if something goes wrong in upload or classification or anything, we can always go to the telemetry file, which is actually in the epitome output folder. There is a log folder in which you will find the telemetry files. So that will tell you if there is any error that happens. So it is often, if you have enough space, it is good to have both data and telemetry. So what is data? Data is basically all the output files that a particular pipeline will give you. And this is the input directory which we drag and drop. After filling all these minimal parameters, we simply do accept and start. Okay, when we do accept and start, it is going to simply upload the data. Okay, so it's going to upload data read by read, and it is going to give you an analysis ID. So analysis ID in this case is 369397. So that is the analysis ID unique for this particular analysis. The cool thing about using epitome agent is that you can start like 10 different analysis at the same time if you want. One can be metagenomic analysis, one can be a 16S analysis, one can be a SARS-CoV-2 analysis. You just simply again go to a new analysis here, and then you choose a different data folder, and you drag and drop that data folder and choose a different pipeline and click upload. So it is only limited by the upload speed of your like the internet bandwidth that you have. If you have really a lot of bandwidth, you can even upload data from 10 different samples and 10 different analysis objectives. And then here on runtime, it tells you how much data gets already uploaded. You see that 29,902 reads got already uploaded, and the analysis in progress. Okay, when you get results, you get this results. There will be a tab which will come available here, which will say results available or report available. You can click that tab and it will take you to our website, epitome.nanoporetech.com. You have to log in using your username and password, the Nanopore registered username and password. And once you log in, you can see the analysis results in our website. And also some analysis output files will also be downloaded to the output folder that you gave while you started the analysis. And this analysis in progress, something to note is that this will never stop, because it is designed to actually think in its mind that sequencing is happening real time. So it is always going to keep checking the input folder for new sequencing data. And because of that, this analysis will not stop for at least a month or so unless you manually go and stop it, because it will always keep looking for new data from the input folder. So, and if it finds new data, it will upload the new data, it will redo the analysis and update your results. Okay. And then all the instances that you are running will be on the left-hand side, as I'm showing here, all the instances that you're running. For example, the FastQ WIMP workflow that we just dragged and dropped started 3 minutes before, and that is the analysis ID. And this is the version number of the pipeline 2021-11-26. And then you see that I ran a human variation pipeline on December 6. And there is another human variation pipeline that I ran on December 6. And I ran a FastQ clone validation pipeline, which is the plasmid assembly pipeline that I ran on December 4. So each and everything has its own instance ID. Suppose some error happens and you don't see any results. When you talk to Nanopore, if you write to us like support@nanoporetech.com, what are the information that you need to communicate with us? You have to tell us the instance ID 369397 and tell us that FastQ WIMP workflow is not generating results for you after 24 hours. You can write to us, and then we will see in the backend if the server is busy or something like that. So, and you see that blue line here or the teal line here, that line tells you which analysis is running, because the epitome agent can actually handle multiple workflows at the same time, right? Because you are simply uploading data. So this interface that you're seeing here is belonging to which pipeline that you're running, so that you can see from that purple teal, the teal line that you're seeing here. So that is the pipeline that is running. If I click this variation pipeline, then it will show the results of that particular pipeline. The analysis is still happening. While this is happening, let's go to the terminal and see what happened to our Promethean analysis. See that we have already completed the bacterial genome assembly already on the Promethean server. Okay, so it says that, you know, it took 7 minutes and 36 seconds to finish the pipeline, and then it took this much CPU resources, and all the workflows got succeeded. And then the results would have already got generated into the output folder. So let me see, I am typing, let me clear the window. Okay, now I'm typing ls, which is list. So there is a folder called output, so which is where the data generally on default goes. So I'm changing directory into the output folder. I'm listing. Okay, now I have a HTML file. So this HTML file is the summary report file that I get from this analysis. Okay, and then I also have a FASTA file which is giving me the actual sequence for the bacterial genome assembly, and then there is this GenBank annotation based on the PROCA annotation, which will give you the gene features for that particular assembly. So I have already copied this data into my Windows laptop, and let me open one HTML report for you and show you how the report looks like for bacterial genome assembly. Okay, so let me go to the let me go here to the training folders, and I'll show you. I have already copied the output folder here. So, okay, so I will go inside and then I will show you. I will double click this report.html file and show you how it looks like. Okay, so this is how a summary report that you will get after you run the Nextflow based pipeline. This is a HTML summary report that you can share with any of your collaborators. And you see that, you know, in that FASTQ files that we uploaded, I mean, not uploaded, that we used for our analysis, there were 5,999 reads and the median read length of 3,480 bases. And then the total megabases in terms of reads were like only 22 MB. So 22 MB of genomic information in terms of bases were there in the reads, because we subset the reads just for the testing purpose, right? So we didn't have enough data because I want the program to run faster. So finally, what I got after doing the assembly by doing this overlap layout consensus by stitching those reads by overlapping one read with each other, and then you get the config, and then which is basically a sum of all. It's a big piece of the jigsaw puzzle, which is the single piece of the jigsaw puzzle is actually your read, and then all reads based on overlap you get the big thread, which we call as a config. And then the largest config size is 0.11 MB, so around 100,000 and 110,000 bases in the genome. So there is a inbuilt reference database comes from PROCA, which against which this sequence will be compared, and it'll give you what is the coverage for that particular config that you got relative to this reference, the best possible reference. And it tells you that, you know, for this 110,000 bases, which is a subset of the genome, because we didn't I didn't supply all the FASTQ files, so that the genome is not fully covered. It only covers just 3% of the genome, but then wherever it covers, it has a coverage of 206x, meaning for every single base that got covered, there are 206 on average reads that cover the single base to give you consensus when you do variant calling and polishing the data. But you see that, you know, the species to which this particular assembly got best match is a Listeria monocytogenes genome, and you see the identity is 100%. As you can see that with a quality score of 99%, and then by doing this assembly and then polishing, if you blast against a reference database and the database of genomes, you get almost 100% identity for the genome that you reconstructed using the assembly. So this tells you that, you know, when you do consensus based calling, I mean, when you do assembly and then polishing, your accuracy of the assembled genome is getting really improved. Okay. And then it also gives you some quality control information, like, you know, the read length distribution of each and every read that you had in your sample. You see that the median is around 3.5 KB, and you see you have some small reads and you have some big reads. And because here you remember we did not set a length filter, so you also have small reads. And then, you know, the median of the read quality score is 10. And then this is actually an alignment map relative to the reference, which is the Listeria monocytogenes reference, and it tells that, you know, 100,000 bases got aligned to the Listeria monocytogenes reference, and then some aligned to the forward strand, some aligned to the reverse strand. That's and then you see the coverage from the coverage is on average 100x for that particular entire region. Okay, and then finally you have the PROCA based annotation, and which gives you the genetic annotation of the piece that we have assembled. And then using the DNA features viewer software, it already gives you this kind of a nice graphical output of what is inside that 110,000 bases that we have assembled. So these are all the genes that are present inside the 110,000 bases that we assembled, and this is the annotation of those genes. You get this nice figure, and then it also tells you all the different softwares that are used in this pipeline, and then the version numbers of those ones. and so on and so on. So this is the kind of report that you get for any pipeline that you run. And then with this, you can have, I mean, an idea of like, every pipeline starts with the QC, and then it gives you some figures. And also not only that, it also gives you the real meat of the analysis, which is your consensus.fasta file, basically your assembly file. The assembly file is this one, subset.metadata.fasta. Because my file name was subset, so meaning that that name got automatically fixed here. So it is if you name your file as E. coli.fastq, then it would have become E. coli.metadata.fasta or something like that. So that is your assembly file, and this is your genome annotation file. So basically inside this you will have the fastq, I mean the FASTA sequence. So you can take and also do blast and things like this, or if you want to use some third-party programs, you can use it to run it. So that is pretty much how you run pipeline using Nextflow. As you see that I ran only a single line of command, and the Nextflow pipeline actually ran through all the list of programs one by one, because this pipeline not only has one single program, it has like 10 different steps, but the Nextflow as a package manager will actually run this pipeline from step 1 to step 10 for you, and then it will summarize the report for you, and it will give you this kind of HTML report. So likewise, we have a plasmid assembly workflow called WF clone validation, which internally will use an assembler called Kanu, and then Kanu will circularize the plasmid and give you annotation of the plasmid based on a plasmid database, and then you can also see the nice graphical view of the plasmid genome annotation. But remember, if you want to do plasmid assembly, you have to start with the rapid kit to actually cut the plasmids and tether the plasmids with the sequencing adapters. So that is WF clone validation workflow. And if you want to do whole genome assembly, you need to use WF bacterial genomes, which uses an assembler called Fly. So Fly is an assembler which is compatible even up to human genomes, but people generally use for bacterial genomes, and people use for genomes up to 100 megabases in size. So sometimes fungal genomes also people use Fly assembler. Depending on the size of the genome, how much number of threads that you want to supply, how much memory you want to supply have to be calculated. Those are additional parameters that you need to apply, and those parameters can be set on your Nextflow config file. Maybe that is one last thing that I will show you. So where to find the Nextflow config files? When Rifki asked me, like, can I run this on a high performance cluster? Yes, you can run this on a high performance cluster, and you can maximize the compute resource depending on how much resource you have for a particular pipeline. How you do that? You can go always to the Nextflow config file, which is always present in the home folder. Okay, so it is inside a folder called .nextflow inside the home folder. And then I list there, you see something called assets. So I go into assets, and then I see epitome labs folder. And then inside that folder, I have the WF bacterial genome. So I go WF bacterial genomes. And then inside that, you see something called nextflow.config file. Okay, so if I go to nextflow.config file, I open the nextflow.config file. Okay, so I open this file, you see there is CPU's number and memory number. So you can change this CPU number. If you, let's say, if you're running in a high HPC, you can, let's say your HPC has like 64 core CPU, and for this pipeline on default, you want to give 8 CPU, you can give 8 CPU. You can change the memory from 8 GB to 16 GB. So because many of the programs do multi-threading, and because of that, if you increase the RAM, the CPU resources, the speed in which it runs and finishes the analysis will become doubled. Sometimes if it is some programs and some steps of some programs are can be multi-threaded, so you will get a faster performance. So that is the use of actually having the Nextflow based pipelines, because these are scalable for high performance workstations. So if I go to the top of the file, it also tells me all the different things like WF, all the basic parameters this Nextflow config file takes, what is the model that it takes for polishing the workflow, and many things that you can, if you want, you can change. I mean, you can also see what is the Nextflow version it is using, what is the WF bacterial genomes software version it is using. All these things are present here. The more the user, the thing that is important for a user is the compute resource and memory resource changing. So if you change this file, then the resource will scale accordingly. So now I'm coming out of this file. Okay, so this is the nextflow.config file present inside the .nextflow folder, which is usually on default goes to the /home folder that you have. So with that, this is one single example of how you can run and launch this kind of Nextflow based Epitome Labs pipeline. So similarly, you can run any pipeline. For any pipeline, if you want the test data, you have to download the actual folder from GitHub. So you git clone that particular pipeline. It will have test data for each and every pipeline. And then using that test data, you can do a test run. You can go to the README file and read the command syntax, copy paste the command with the, and give the test file or test data file as your input folder. And then you can run and get some results like how I showed you for the bacterial genome assembly. So let us go to the Epitome agent and see whether we have results. Yes, we have results. So for the metagenomic assembly, you see that it already generated results for you. Perhaps it took 15 minutes ago, we started the analysis. Now we already have results. You see 57 megabase of data got uploaded. And then you see that, barcode 1, barcode 2, barcode 4, like, different barcodes. Data is getting uploaded. Not everything got uploaded already, but you will see some results for whatever data that got uploaded until now. Okay. So let's see what it is showing us. Okay, so it is still not showing us. I mean, probably just finish the QC part of the workflow, because every workflow on the Epitome agent also has a QC tab. So because you want to know, data sanity, and here it tells you that, out of the 20,000 reads that got uploaded, only 17,924 got filtered, because you remember we set a Q-score filter of 8 and we set a read length filter of 500 bases and above. So some reads got filtered already, and then it tells you 2,711 reads got filtered based on QC, and some so that way, so the remaining data gets analyzed, and that is the 81.8%, which are all the data that comes after filtering. And then it gives you for each and every barcode how much data got uploaded. For barcode 1, 9,000 reads got uploaded, barcode 2, 9,500 reads got uploaded, and things like that. And then finally you will have when the analysis is over, you will have in this WIMP tab, you will have the sequence taxonomy classification. So far it hasn't generated the sequence taxonomy classification. It may take some time. And this is the fun about Epitome Labs, because as and when a little data gets uploaded, that data gets analyzed, and whatever analysis got completed, it will show you. Okay, so if you keep refreshing the file, you will know that if more data gets generated. And finally, you can also do a print PDF report of the summary file here, and it will also download some like output files to the output location that you gave. With that, this is one snapshot of how you can analyze our data using our Epitome agent interface. The other snapshot is how to run the Epitome Labs workflow on a terminal. So with that, I think it's a nice mix of what is possible with Oxford Nanopore, and you can try different flavors with test data. You can always download test data from our website called OINT Datasets. We are lucky. We already got the WIMP data here loaded. You see, it's 100% bacteria out of 10,000 reads. It's close to 100%, which is bacteria. And let's see what these are. You see, some are E. coli, and some are Klebsiella, and some are Acinetobacter. If I click that, it tells me that 160 reads actually map to Acinetobacter, and then it tells me 4,636 reads actually map to Escherichia coli. You see the taxon here. The taxon here is set at. If you see this taxon, it is set at genus. So you can always change it to family, then it will get a higher thing. If you set it to species, you will get a different, it will be segregated based on the species. And then this is a taxonomy tree. This is not a phylogenetic tree, but it's a taxonomy tree, which gives you the classification. And then the thickness of this line basically means that many reads support that particular clade. And then the thickness size of the node also tells you how many reads actually map to that particular clade. So you know what is the predominant species. But one thing I have to mention is that, these are single read based analysis. So it's a single read match against the database using a centrifuge algorithm, and then basically based on the read classification, you get the assignment. And then there will be a CSV file that gets auto downloaded, and then this CSV file will have for each and every read what was the assignment, what was the tax ID, and what is the taxon classification, etc. You will have that in the CSV file. and you see the minimum abundance cutoff. For example, if you want to only see the top 3% of your species, you can click this, and based on read abundance, it will only show you a simplified taxonomy tree, which only has the top 3% of the species that you want to see. So this is how you can actually use the epitome agent for your real-time analysis. On the other hand, you can use the epitome Nextflow for your production scale HPC environment analysis. With that, I will stop my practical demonstration. If you have any questions, we can follow.
Thank you, Bala. So I think we have question in the chat.
Yes.
So you can read.
Franz from Biomedical Research BRIN. So can we execute epitome pipeline in Windows subsystem for Linux? Yes, you can actually execute epitome labs pipeline for using Windows subsystem for Linux. And if you download the latest version of epitome labs, is called epitome labs 4.0, version 4.0. Now with version 4.0, which we released like last 10 days before, if you try to download the software and install that software, it will automatically see whether your system has WSL2 environment, and if it doesn't have WSL2 environment, it will show you additional requirements are needed for this software to function, and then you can follow the steps to actually install WSL2 through the Epitome Labs interface. Alternatively, you can also install, if you have pre-installed WSL2 interface, you also need Docker desktop, which is another requirement because all these Epitome are Docker images. So you need Docker desktop, and then you need the Epitome Labs software, and plus you need the WSL2 backend. And with that, you can actually launch whatever I'm showing you in the Nextflow interface through the command line. You can launch through the Epitome Labs graphic user interface with the WSL2 backend. But just to add to a note that, I mean, WSL2, because of the WSL2 has limited support for raw socket communication. So it is in terms of efficiency and in terms of, I mean, the quality is the same, but sometimes performance and I mean, it is not that straightforward when compared to a virtual Linux environment. WSL2 will need a little bit of extra effort because there are so many moving parts with the Windows WSL2. It is not really exactly a Linux operating system. So that way, running inside a WSL2 environment, sometimes some novice users may find it difficult.
The second question is from Bu Ernest or Pak Ernest. Hello, you can read in the chat.
Let me see. Yeah, Ernest, we just got Nanopore Menayan and we'll do training in February. We don't have experience about this test, but it looks very interesting and challenging. Could you give me tips to study the basic test? If you have experience identification bacteria from product probiotic. Yeah, I mean, many people run, metagenomic sample using the what's in my pot workflow. And what they do is that they start with the Zymo. For example, Zymo is, if you're from Zymo, you can download a known species of bacteria, let's say 10 species, 20 species, and you can actually take the Zymo DNA and do a library preparation with a kit, and then upload the data and see, because the Zymo DNA has a known concentration of a certain species, you can see whether you can recover all the species with all its abundance, and that way you can kind of quality control whether everything is working nicely in your hands, and then you can run your probiotic sample. So for example, when you're running this kind of metagenomic analysis, right, so you sometimes do not know how many bacterial species or microbial species you have virus, you have so many different things, and you may have like thousands of species in your sample. So if you really want to find even the species which is present in like 0.01% in frequency, then you need to actually sequence that much. You have to sequence so much more to actually find the really lowly abundant sequence. So there is no finite limit for how much you can sequence, because when it comes to metagenomics, some people even run metagenomics on a Promethean flow cell, because if they really want to find some samples which are really niche and very small frequency sample, then you have to run the sequencing for a much longer time on a bigger flow cell. But if you're looking for some predominant species, you can run it on a smaller flow cell, like a Menayan flow cell, which can even generate 30 gigabytes of information for you. And there are several publications already out there which uses the WIMP database for their analysis, and you can always check in PubMed how they have done it, and how people use WIMP for their analysis. Okay, Ruby. So yeah, go ahead.
Okay, thank you, Mr. Bala.
Yeah.
Such a nice presentation.
Thank you.
I want to ask about how, is there's any criteria when we choose the assembler? For example, few days ago, I assembled my Promethean result using Fly.
Okay.
And it doesn't give any context.
Okay.
Even the,
so the thing is, yes, the common problem with Fly is that Fly has two issues. One is that it needs some optimal coverage. What is an optimal coverage? So something like a 30x to 60x to even 150x. Fly understands it as an optimal coverage. Suppose the problem with bacterial genomes is that sometimes you over sequence bacterial genome because bacterial genome is just 5 MB. If you have like 2 GB of data, then you have 400x coverage for a single genome. So, but then if you have 10 GB of data, then you have like 2000x for a single genome. So when you have that much coverage, Fly algorithm kind of cracks. So that is one technical issue with the Fly third-party assembler. And also when you have really small assembly, like example plasmid assembly, which is just 5 KB, 10 KB, something like that, Fly also struggles to assemble really tiny genomes because it has some heuristics to actually join the initial pieces, which we'll call as a disjointing. When it does that stage, it needs some pieces. So if it is like, a single nanopore read can be as big as 10 KB. So it has nothing to really assemble because your genome is already only 10 KB. So in such cases, Fly can fail. The third reason where Fly can fail is that if you don't have enough RAM, Fly can give you a signal term -9 error, which is basically a memory error, which you can find from the log. There is always a nextflow.log file where you will find why some program stopped if you are using a nextflow based pipeline. And then with that, you can find out what is really wrong. So sometimes you may have to tune the parameters depending on the size of the genome. So Fly also takes the approximate size of the genome that you feed in, and then based on that, it does some calculations. So for really small genomes, the suggestion is you can go and use our WF clone validation workflow, which uses Kanu assembler. Kanu is a gold standard assembler. It can also assemble really small plasmid genomes, and it uses at the tail end, it uses a software called Tricycler to circularize the plasmid assembly, which is coming from Rianvik's Rianvik. So you can try the WF clone validation tool if you are actually working on really small genomes. But if your coverage is really high, you have to subsample your coverage to a, like for example, the subset.fastq, you just only randomly subset your fastq file to a coverage which is in the range of like 100x to 200x. So that way Fly wouldn't really crash.
Okay, so it depends on the size of the genome.
Yes, it depends on the size of the genome. For example, if it is an E. coli genome, let's say 5 MB, then you can input a data which is like 500 MB in space, for example, 500 megabases. You can input up to 500 megabases, which will give you an average 100x coverage. But if you input 5 gigabases, then it is 1000x coverage, then Fly will crash because of I mean, it cannot really do the disjointing when it has so much data. It has some technical issue.
Okay, and second question for the polishing program. I read some paper that they use about two polishing programs such as Raccoon and Medaka.
Yes.
How when you need to do multiple polishing?
I mean, Fly comes with Raccoon, and then if you use our WF bacterial genome pipeline. So the cool thing about actually using nanopore recommended pipelines is that what we have optimized the pipelines for what are all the kinds of tools that needs to be used. For example, whenever you have a pipeline from us, I would suggest or recommend that you can use our pipeline, because we have optimized how much policing you need, we have done so many data analysis at the back end to know how many policing is needed. One round of ragam and one round of medaka is a general consensus that many bioinformaticians follow. And we have incorporated that in our WF bacterial genomes pipeline.
Okay, thank you.
Yeah. Okay, let me check in the chat if there is any question. Okay, so there's not much question on the chat. Okay, I mean, we are not limited to just questions here. I mean, you can always go through Rifki and feed forward the questions to us. I mean, you can always communicate indirectly or directly through support.nanoprotect.com. It is an ongoing exercise, like what Rifki pointed out. If this exercise, if you find useful, we can run this kind of modular exercises for different projects, like, WF Human Variation Project. And you can also feedback to Rifki, like, for example, what you need more. Do you like more terminal-based real-time practical, no PowerPoint, just directly analyze data and show in real time? However, like you want, I mean, like the majority speaks, I can customize training for you accordingly. If you have a lot of human genomes to come in the future, we can do something like that. Like, we can do also metagenomic specific workflows, like more metagenomics in detail. And also one more thing that I would like to encourage you is that there is something called Nanopore Learning from our community webpage, Oxford Nanopore community. Everybody who has a Nanopore device or some products from Nanopore, you have a login with the Nanopore website. You go into Nanopore website, and then there is something called a Nanopore Learning webpage. And if you go to the Nanopore Learning webpage, we have a lot of videos. There is Bioinformatics 101 videos. There is videos for metagenomics, entire metagenomics course, which will cover both from library preparation to data analysis and all the software tools that we use. We are developing a course for human variation. So likewise, we have several courses. You can also learn from there. And on top of it, if you really want to see practical analysis, I can also jump in when we organize with Rifki.
Yes, thank you, Bala. Jadi Bapak Ibu, kalau terkait dengan Nanopore Community, nanti mungkin kita bisa undang ya, supaya bisa login ke website Nanopore dan join Nanopore Community. Di sana banyak info ya terkait dengan bioinformatics, dan juga kalau wet labnya juga ada gitu ya. Nanti mungkin kalau ada yang perlu bisa ditambah sebagai partisipan yang dari BRIN. Ini untuk internal BRIN. Nanti kami bisa invite melalui email, sehingga nanti bisa bisa akses ke Nanopore Community. So Bala, so we are in BRIN right now. Is at the beginning stage of having a sequence program, and right now is doing the sequencing for the collections, microbial collections, plants and animals at BRIN. So actually, we would like to have environment where from the front to the end we want to store the sequence results at our own storage, and then people can do analysis directly from that storage.
Yes.
Using the computing resource that we already that will be installed in this month. So I think what we want to have is, like, we would like to create the system or the environments at the SPC system, like Nextflow module or Bioinformatics module, like that. Then after that, the environments are already been installed. We would like to have some kind of offline for people. Maybe you can join online, but people who want to do analytics more, I think better to have like offline workshop, and with the specific issues like, for example, just for plant or just for animal or human or so on. So I think this is just beginning, just opening to the audience here from BRIN to do research in genome or related to it.
Sure.
And we are in the Research Center for Computing. We are preparing the environment also. I mean computing environments. That's why I think we need to, maybe we need to consult to you how to, like, for example, providing what modules and so on.
Yes.
So people can optimize.
I will follow up with you or Shyam, and then I mean I will give him the instructions for installation of the modules, necessary modules that are necessary to do this. And if there is any trouble, I can jump in Zoom call, or I mean I can also jump into your HPC and then see if there is any issue, and we can do some testing of how the like whether the program successfully run on your HPC, and then we can leave it for users. If you I mean I can align with whoever you want me to align with, and give them the instructions to set it up.
Okay, I think if from floors there are questions, you can raise your hand and directly open mic or type in the chat. So I will count to 5. If not, then we can conclude the meeting today.
Ada 1, Pak.
Ya, itu oke. Ya, Bapak atau Ibu ya, Bu Ernest, kayaknya Bapak ya. Ya, nanti kami akan tetap kontak ya, kalau ada acara juga kita publish. Intinya sih kita HPC sistem di BRIN juga sifatnya open. Jadi kalau nanti Bapak Ibu yang dari non BRIN ingin menggunakan juga bisa, asal untuk riset ya. Jadi tidak boleh untuk yang lain. Terima kasih atas komentarnya. So I think this today is just opening for our next meetings, but I'm still don't have fixed schedule, but we will inform to you because we are now also in the building of the sequence lab at BRIN, and this Oxford Nanotechnology is just one of the sequence machine that we have, and we would like to have implementations so people can do analysis and also deposit the sequence in the BRIN systems, in our system. So thank you, Bala, for your presentation today. Very nice and helpful. I hope it's not discouraging people who doesn't have a limited knowledge about the bioinformatics or command line, because we have to learn continuously and don't be afraid to command line instruction, because it can be learned actually for everybody. So thank you for the audience for attending this meeting or workshop. Thank you for Bala. We will meet maybe in the next month, but I don't have fixed schedule yet. Before we closing, I think from me as moderator, I would like to say thank you to the presenters, Bala, and audience, and apologize if I made some mistakes on delivering this session, and I will give the session to MC Bu Maulida, please. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.
Yeah, I also want to thank everyone for organizing. This is a wonderful exercise, and it's a continuing thing. And I totally acknowledge that and recognize that we will give you continuous support as much as we can. Yeah, so definitely we are with you in your sequencing exercise until you get your data. Yeah.
Okay, thank you, Pak Rifki, Mr. Bala, for very nice presentations. And to close our sessions today, let's take a photo together. So for the participants, please open your video. I will take your photo. Okay. Are you ready? Yeah, okay. I will take the photo. Start from slide 1. 1, 2, 3. Okay. And the next slide. One, two, three. And the last slide. 1, 2, 3. Yeah. Okay. Thank you for attending our workshop, and see you on the next workshop. Wassalamualaikum warahmatullahi wabarakatuh.
See you, everyone. Have a good
See
day.
you.
Everyone.
Bye.
Bye bye. Thank you for.
Bye-bye. Bye-bye.
Dari total anggaran dari total anggaran